All right, welcome everybody. My name is Dr. Nikki Whitley. I'm from Fort Valley State University in Fort Valley, Georgia. We're going to be hosting the last session of our weekly worm webinars, which is a collaboration between my university, University of Maryland Extension, Susan Shanian, Kwame Matthews at Delaware State University, and our presenter today, Dr. Delia O'Brien from Virginia State University. Dr. O'Brien?
Okay, thank you, Dr. Whitley, and welcome everyone to today's webinar. Today I'll be speaking about selecting worm-resistant animals. So essentially, in the wake of increasing drug resistance, we've had to figure out alternatives for effective worm control in our flock and in our herds. Some alternatives include targeted selective treatment, pasture management, nutritional supplementation, and genetic selection. It's important to note that no alternative by itself will likely be effective, and that you really need an integrated approach to control worms effectively on your farm. So choosing for resistance or resilience is an important strategy, and it can go a long way in helping you find and selecting the best animals for long-term health. It's going to help you to build a more resistant herd or flock. And so today's presentation will focus on how best to choose those animals with these traits to help effectively control internal parasites. So as I mentioned before, resistance and resilience are the two major concepts relating to an animal's ability to limit or withstand a worm infection. Resistance is the ability of the host to limit infection. They will have a natural ability to suppress establishment of parasites or worms inside their body. So this suppresses their egg-laying capacity, and essentially they expel the adult worms. And the way that we've traditionally identified these animals is by looking at fecal egg counts and looking at the results of those. An animal that is resistant will present with no clinical signs and will have fecal egg counts that are low whenever they're evaluated. On the other hand, resilience is the ability of the host or animal to withstand a challenge or infection, and they're still able to perform. So this is assessed by taking your pack cell volumes or FOMACHA scores. It can also be assessed by body condition scores and other performance indicators. These are the animals that as I mentioned, they have no clinical signs. However, if you assess their fecal egg counts, those might generally be high, even though they're showing no clinical symptoms. So as colleagues, from time to time, we've had discussions about which one is better, resistance or resilience. I, for one, believe that parasite resistance is better and more important. And this is because resilient animals, even though they're productive under parasite challenges, and they do require less deworming, they tend to spread a lot of parasite eggs onto your pasture. Okay, and this could be causing issues in more susceptible animals. So even though that high parasite load is not affecting them, it could be affecting or causing issues in your more susceptible animals. On the other hand, you have your resistant animals, which, as I said before, I prefer. Resistant animals, they also reduce the need for deworming, but in addition, they reduce pasture contamination. So they're not letting out a lot of eggs onto that pasture and contaminating more susceptible animals. And the most important part is that resistant animals actually pass their resistant genes onto the next generation so that the younger animals are also more resistant. So in my opinion, resistance is more important than resilience, but there are some folks that might disagree. And I believe my colleagues will also agree with me that resistance might be better. In the previous slide, I mentioned that resistance can be passed on from one generation to the next. So this leads us into heritability. Heritability is essentially just a portion of a trait variation in a population of animals that are due to genetic variance. So both parasite resistance and resilience are heritable in their quantitative traits, meaning that they're controlled by a number of different genes. Parasite resistance is considered to be a moderately heritable trait, with heritability estimated to be between 20 to 30%, and parasite resilience tends to be a little less heritable. Selecting lines, what this all means is that you can select lines or individual animals that have an improved ability to regulate their parasite populations, and overall this will increase the immunity against parasites at the herd level. So using parasite resistance or even resilience, they can help us to select individual animals on our farm, and by selecting for this, then we can increase our overall immunity against parasites at the herd level. Before we go any further and talk about steps to select animals, we have to talk about host immunity. So we're all aware that there's some animals on our farm that are more susceptible than others to internal parasites, and then some that are less susceptible. Our most susceptible animals are going to include our young animals, or kids, or lambs, crias in the case of camelids, or periparturient females. We all know that there's a lapse in their immune system around the time of lambing and kidding. So these will be your most susceptible animals. And any old animals that you have on your farm. Whereas your less susceptible animals are going to be your open females, your bucks, and at times, and your pets could also be less susceptible, just because we tend to have, if they're for pets, we have less animals and just less contamination because we don't have that many animals, and they're fed better than or commercial herds. One of the key factors in determining whether an animal can resist or allow parasite infection to establish is that host immunity, and this is controlled by the animal genotype. More effective immune response is going to result in low larval establishments or worm egg shedding on pasture. There are two types of immunity that we see. There's innate immunity that's present at birth, and there's also an acquired or adaptive immunity that develops as animals are exposed to parasites. So throughout the last couple of weeks when we've been talking about the control of parasites, we're not urging you or telling you that the goal is to have parasite-free animals. We need animals to be exposed to enough parasites so that they're developing natural resistance to these worms. We're just not trying to expose them to too many parasites so that their systems become overloaded. Young animals they start to develop immunity to parasites slowly with exposure over time. Lambs tend to acquire immunity at an earlier age than goats do. They acquire immunity at around 4 to 9 months of age. In goats, this could be longer, at or greater than a year old, and some really never acquire that complete immunity. Of course, immunity is going to be breed dependent. We know that some breeds of animals are more resistant to parasites than others. To develop this immunity, they do need regular exposure to internal parasites. So again, the goal is not to keep them parasite-free, it's to expose them to enough parasites that they're developing or gaining this natural immunity to internal parasites. So if they're not exposed to those parasites, then they could lose that immunity. When we're talking about adults, they tend to remain relatively resistant to infections. So once they've gained that natural infection based on exposure during their younger years, they tend to remain relatively resistant. And the only times you'll see an immune response is at times when their immune system is suppressed, and this could be due to any normal stressors such as low nutrition, any disease that might be in the herd. We already know that high-producing females, those that have multiples, they're heavy milkers, those types of females, that production suppresses their immune system and might make them more susceptible to parasites at those times. There are some natural ways of boosting immunity in or herd or flock. One of these is just not placing very young animals on heavily infested pastures. Again, we want to expose them to parasites so that they're gaining this natural immunity to parasites, but we don't want to overwhelm their systems by putting them on heavily infested pastures. Ensuring quality nutrition. We've emphasized this throughout all our presentations that nutrition is extremely important to promoting good immunity and helping these animals deal better with parasite infections. Separating and feeding females based on their litter size. The more, if they're high producing females and have multiples and are heavy milk producers, then they're going to have more of an nutritional demand. And if we separate and feed these females, then we're providing adequately for these animals. And any reduction in stress, such as using low stress handling techniques, will help to boost that natural immunity in these animals. Now, looking at breed resistance, we've all heard, or we all know that there are some breeds that just have been naturally selected for resistance to internal parasites. And these breeds were developed in situations and climates that favored internal parasites, and essentially were selected by natural selection, survival of the fittest. And we all know of some producers right now who just, they do that. It's a survival of the fittest kind of operation, and they throw animals out there, and whoever survives, that's it. And this might seem like a inhumane or sort of crude method of doing it, but it's effective at picking out those animals that are more resistant. So these breeds have been identified over time as being just more naturally resistant to internal parasites. And the way that we've traditionally identified these animals is by looking at fecal egg counts. So remember that I mentioned before that resistance, that natural ability to limit infection, is assessed by fecal egg counts. So we identify these animals or these breeds based on fecal egg counts. And that has allowed us to be able to identify some breeds such as hair sheep breeds. We know that St. Croix and Barbados black belly hair sheep are highly resistant to internal parasites. Katahdins don't tend to be as resistant as, say, as St. Croix. There's more variation in fecal egg counts across the breed, but compared to other breeds, they do tend to have natural parasite resistance. Research has indicated that Texels might be more naturally parasite resistant. Gulf Coast native, Florida native, those breeds, as we talked about, natural selection, survival of the fittest, tend to be more parasite resistant. Hog Island, even though that's unsure if it was just based on isolation or just natural parasite resistance. Some goat breeds that are considered to be more parasite resistant would be your Spanish breed, your Kiko breed, and your myotonic breeds. And there's been some discussion about Savannah, but there's insufficient data out there or inconsistent data out there that has shown this. But for most of these breeds that are listed, such as your St. Croix, there's enough data out there that shows that they tend to be more parasite resistant than other breeds that are out there. One study that was conducted in looking at meat goats at Tennessee State University, they compared reproductive growth and health traits among specific breeds, the Boer, Spanish, and Kiko. And what they found was that annual rates of parasitism were greater for Bordeaux compared to Kiko and Spanish does in that study. And they also found that postpartum fecal egg counts were also less for Spanish and Kiko dams compared to Boer dams. There is limited data out there comparing, especially goat breeds. They're more comparing hair sheep breeds versus wool breeds. This is one study that's out there that has compared goat breeds and have found that Kiko and Spanish breeds tend to be more parasite resistant than Boer goats. So a lot of you that are out there probably aren't raising animals in a purebred system, and you're probably doing some crossbreeding system. Like we mentioned that Katahdins are generally a parasite resistant breed, or St. Croix, one of the most parasite resistant hair sheep breeds. But typically these breeds, even though they have that parasite resistant trait, aren't generally noted for their growth and carcass traits. And so if you're doing a commercial sheep production system and you want to have better growth or better carcass traits, then a lot of folks do a crossbreeding system, which is simply just mating animals with different breeds. And you'll see farmers that have a maternal flock of Katahdins or a maternal flock of St. Croix or Kikos or Spanish does, and then they'll cross these with a sire that has that growth and carcass traits that you're looking for. And these just crossbreeding just takes advantage of breed complementarity, where it balances the strengths and weaknesses of different breeds. So you get that natural parasite resistance from your maternal breed, and then you're crossing it with a male that has that growth and carcass traits. And crossbreeding also allows for hybrid vigor or heterosis, where it's a superiority of crossbred animals as compared to the average performance of their purebred parents. So this is simply saying that those offspring, those crossbred offspring, typically do better than the average of both purebred parents. This table right here depicts some results that we got in a study conducted during my dissertation at University of Maryland Eastern Shore, where Dr. Whitley was my major advisor, and Susan Shanian served as an advisor on this project. But essentially, we compared the growth and carcass traits of Katahdin, Dorper, Texel, and Suffolk sired Katahdin offsprings to essentially see the performance. We're focusing mostly on that fecal egg count data. So if you look a couple of rows down, you'll see that day 90 or weaning fecal egg count and the day 120 fecal egg counts. And you could see that average for the Katahdin purebred, the Dorper by Katahdin cross, the Texel by Katahdin cross, and the Suffolk by Katahdin cross. And you could see that the overall averages for the Katahdin purebreds and the Texel by Katahdin crosses were lower compared to the Dorper sired Katahdin and the Suffolk sired Katahdin. So this is what I was talking about, where you have that purebred system. Katahdins are known to generally be resistant to parasites, but the Texel breed has also been shown to have some parasite resistant traits as well. And this data has been supported by Virginia Tech research. And Dorpers, even though they there were rumors or there were folks that were saying that they're a more parasite resistant breed, but they tend to be more parasite resilient, a more parasite resilient breed. So they are a hair sheep breed, and they do have faster growth weight and better carcass traits. However, they tend to be more parasite resilient. They don't show clinical signs but have higher fecal egg counts and not naturally parasite resistant. So selection based on a breed can be an effective tool in reducing your worm loads. Especially in a purebred flock, you could choose parasite resistant breeds in your operation, and that will be an effective tool in reducing worm counts. However, within every breed, there is a wide range in fecal egg counts, and some individual animals will always do better than others. So it's advisable to evaluate individual animals based on their own merit, because you'll have that wide range in fecal egg counts and just overall performance. So we always want to identify those animals that have lower numbers of worms. As I mentioned, there's a distribution, a wide variation in fecal egg counts in your herd. Those more parasite-resistant animals will decrease the overall need for deworming. And if you can identify those animals that need deworming, then you can cull out your parasite-susceptible animals, and that will increase your overall flock resistance, reduce pasture contamination, and decrease the deworming frequency. You've all probably heard about the 80-20 rule, where essentially 20% of the herd or flock shed 80% of the parasite eggs onto your pastures. So if you can identify that 20% of the herd or flock and cull those animals, then you'd have gone a long way to helping to reduce your pasture contamination and decreasing your deworming frequency in your herd. The most accurate way, as I mentioned before, to identify individual animals that are parasite resistant is to conduct fecal egg counts. All right, resistant animals will have low fecal egg counts. They're not going to shed that many eggs in their manure, and they've got a strong immune system, or developed a strong immune system in response to their exposure to parasites over time. And this is going to be your most effective. Looking at their fecal egg counts is going to be your most effective way of looking at or selecting your parasite resistant animals. When using fecal egg counts to select those parasite resistant animals, the accuracy is going to increase with multiple samples. Say, for instance, you have an animal that you like how it looks, like how it's growing, and you want to decide should you select that animal, and you're looking at fecal egg counts. It's best to take samples, multiple samples from a similar age range of animals during the same season of or year. So if you want to select a male, say one of your bucklings or ram lambs, then you would select multiple samples from that group so that you could do a proper evaluation from that group, not just from that one animal you like, because you want to compare it and see how that animal is doing. Information from fecal egg count is useful, and as I said before, we cannot overstate this. It is the best assessment of resistance in a flock or herd. Now, you can learn to do your own fecal egg counts. And a few weeks, I believe it was the second weekly worm webinar, we did do a fecal egg counting primer. So we have that video available for doing your own fecal egg counts. But the best way to learn to do your fecal egg counts is to actually go to the lab of someone who does it, or meet with the producer who's been doing it for a number of years and very familiar with it, and just count behind them to see if you're getting similar counts, because there could be a lot of debris and other material that are in the fecal sample, and you want to make sure that you're counting eggs and you're correctly doing this. But you can learn to do your own fecal egg count so you can do a good assessment of your herd. Of course, fecal egg counts are offered by many veterinarians and labs that are out there. If you go to the wormx.info website, there's a list of labs that are doing this at, not high cost for producers. They cost about $5 per fecal egg count. And these are West Virginia University, I believe, Texas A&M, and Jim Miller. And if you go to the wormx.info website and just look at labs that are doing cost-effective fecal egg counts, you can find where these locations are, and it tells you how to collect the samples and how to ship them if you're interested in doing that. But also be aware that there are other veterinarians out there that do it, and other labs, but the cost range is going to be different. If you do this yourself, it's depending on how many you have to do. It could be labor intensive and costly. It's just basically initial costs, because you have to get a microscope, get the master slides, but you can make your own fecal solution for doing your fecal egg counts. And the master slides will last you a very long time, as well as the microscope. But you can learn to do these on your own or use a lab. And the best way to use fecal egg counts is to measure and record it during the parasite season. You're not going to measure fecal egg count during the off season and expect this to give you any vital information. It has to be measured and recorded during the parasite season. And you want to take multiple fecal samples, at least 2 and at least a month apart. And it's important to do these fecal egg counts, especially when you're selecting potential sires, because for a number of us, that sire, we have single sires in our breeding groups, and that sire will contribute 50% of the genetics to those offspring. And so it's very important to do this, especially when selecting our sires. And small results between fecal egg counts are not necessarily meaningful, but those large differences will indicate those animals that are more resistant or not. As I stated before, if you're looking to select pasture-resistant animals out of your kids and lambs, then you wanna make sure that you're using contemporary groups. So same age, same management, everything. And you're gonna collect fecal samples from a number of these animals, and not collect from those that were previously dewormed, because those are gonna have low fecal egg counts, and you don't want that to affect your data. But then you're gonna collect those fecal samples, do those fecal egg counts, and you're gonna multiple fecal egg counts over that parasite season, and then you can look at those numbers and select those animals that have the lowest fecal egg counts. And you're not going to want to just look at those fecal egg counts. Remember, you're not going to try to forget about their growth performance and so forth. So you're going to look at their growth performance as well, and then use fecal egg counts. to support that. So I believe this was from a past meat goat performance test, but I'm not sure. But this table represents the kid ID, their fecal egg counts taken at two different points a month apart. And if you look at their fecal egg counts, and we're trying to look at this to identify animals that are more resistant or more susceptible to parasites, remember again, important to compare them as a group and not as individuals. So we can see if you look at KID ID 103 at the fecal egg count done in June indicated that there were no eggs present, and fecal egg count in July just indicated a low fecal egg count of 150 eggs per gram. The same thing for 111, low fecal egg count at both times. And if you look at Queen and 105 and 112, you'll see that these had higher fecal egg counts at both time points. So again, this table shows you a great way of going through and looking at those fecal egg counts and trying to identify those animals that have low fecal egg counts consistently. And this will help you in selecting for your parasite resistant animals out of your herd. So a lot of us are probably doing selections already based on the Falacci system. So for those of you who are not familiar, the Falacci system is just a tool for determining those animals which need to be dewormed or not. And it's based on the degree of anemia by giving a score of 1 to 5 on their eye membrane, with 1 being bright red and healthy and 5 being pale and basically severely anemic. And it is an evaluation of the barbapole hemoncosis infection in these animals. We know it was previously developed in South Africa, and most of us are using this on our farm to determine the need for deworming or not. It's a simple and inexpensive tool, so even if you're not getting fecal egg counts done regularly, you should at least be doing your Falacci scores, and more importantly, you should be recording those animals that have high versus low fecal egg counts and those animals that need to be dewormed multiple times throughout the year. As I mentioned before, those resilient animals will have a good Falacci score even though they have a high fecal egg count. So even though you're doing Falacci on your farms and you're recording this and you're using it to determine their need to deworm, in order to truly differentiate between your resistant and resilient animals, you should be supporting these Falacci scores with fecal egg counts. Okay, and this is the only way you'll be able to determine true resistance versus resilience. Remember, if you're going through just looking at clinical symptoms, whether you're using just the Falacci score or using the 5-point check or other performance indicators, a resilient animal will not show clinical signs. So it could have high fecal egg counts. So when you're selecting based on good Falacci scores, you're selecting both parasite resistant and resilient animals. So a good tool to support your selection decisions is to also have fecal egg counts to back up that Falacci data or that deworming frequency data. So I mentioned the 5-point check, and this slide just tells you what the 5-point check is, and it addresses limitation of the Falacci by determining the need for deworming for all internal parasites. So it not only looks at the anemia of the animal, but it also looks at all these other traits, such as it looks at the fecal swelling, it looks at body condition score, it looks at that coat condition or the nose and bottle jaw. So it's a very good tool in addition to the Falacci in our targeted selective treatment approach to deworming on our farms. Hopefully most of you are not only going through during your parasite season and evaluating your animals weekly or every 2 weeks for your Falacci scores and just determine the need to deworming and not writing this somewhere or not putting this down somewhere. Ideally, we should be recording all these data. So if you're going to be doing your Falacci scores or doing your 5-point checks every week or every 2 weeks, you should have a sheet with you that you're recording their names and you're recording their Falacci score. You're recording if you had to deworm or not. And at the end of the year, going through and looking at this data in addition to other data such as for your females, how many offspring they had, their overall performance, and so forth, and basically selecting based on that. You should be recording these data. And so you want to cull those animals that need multiple dewormings a year. Of course, we're going to take into consideration if they had to be dewormed based on high stress, stressors that couldn't be avoided, multiples, high lactation, disease, or something else. You want to take that all into consideration. But if there's an individual that needs multiple treatments each year, then those are animals that you don't want to be keeping in your herd or flock. And then you don't want to be selecting any offspring from these animals that require frequent treatment. Remember, I mentioned that parasite resistance is a heritable trait, so they pass it on to their offspring. So we should get into a practice, and we all know it's difficult, especially when you have these pets. It's very difficult sometimes to get rid of specific animals from a herd. But really, it should not be just based on how they look or how much we love them. It should be based on data and including or deworming frequency. And Fibonacci score data is important to selecting and make sure we're trying to get towards a parasite resistant herd or flock. In our young animals, it's also important to record the weights of those growing animals so that we could select the animals that do best under our own production systems, and then combine this with their fecal egg count and Fibonacci scores so that we're picking the best animals that will do well under our production system. So the overall goal of doing all this is that we want to select the best animals. So the male represent half the genetics in the flock or herd, and as I mentioned before, most of us have one male that we breed to our females, and he influences the genetics. He's going to contribute 50% of the genetics to your offspring. And so you want to make sure that you're selecting the best male for breeding to your females. And if you're selecting or saving replacement females or replacements, you want to make sure that you're using all this data to select the best. Why would you say, for instance, you've already invested in a St. Croix maternal breed of ewes or Spanish or Kiko females, and why would you then breed them to a susceptible or unknown male? You'd want to breed them to a male that you know has parasite resistant traits, and this can help to provide more resistant or pass more resistant down to those offspring. So our selection should be including parasite resistance when we're going through and trying to decide. And then we want to cull the worst animals. This cannot be stressed enough, especially those females. They only influence the genetics of 1 to 4 offspring per year. We want to go through and we want to identify those females that are more susceptible to parasites, get rid of them, and just keep those that have good parasite resistant traits. And again, keep in mind that your higher producing females, yearlings, younger animals, they're more likely to have high fecal egg counts and require deworming. So we have to take into consideration the entire picture and not select or choose solely based on one criteria. So the only way we're going to know all of this is if we keep good records in our herd or flock. The National Sheep Improvement Program, or NSIP, they calculate what's known as EBVs, or estimated breeding values, for sheep producers and breed associations in the US. EBVs are science-based, industry-tested measurements of heritable traits that can be tracked and measured, and they have been proven to improve on-farm productivity and enhance breeding decisions. So simply, these are proven methods that accurately predict if an animal will pass on important traits or not. And for today's talk, we're focusing on parasite resistance. So NSIP collects data for other production traits, but for today's talk, we're focusing on EBVs for parasite-resistant traits. Currently, Katahdin and Polypays, they have significant fecal egg counting data for EBVs for parasite resistance through NSIP. However, any producer, regardless of breed, regardless of species, goat producers can do this too. You can submit fecal egg count data, and they usually recommend for fecal egg count to submit a weaning fecal egg count data and post-weaning fecal egg count data to calculate these EBVs. So what EBVs allow us to do, it helps us to identify the differences that are due to genetics alone. Okay, and so it not only takes into consideration that individual animal, but it also looks at data on related animals. And it can do this within the same flock, and it can do this in other flocks as well. So this is a proven method or a proven standard for determining whether an animal will pass on these traits or not. So if you're buying males, remember we said for your males, select the best. So if you're going to select the best, then you want to purchase a male that's either been in a performance test and you have that fecal egg count data. But the best thing to do is purchase males that you know have estimated breeding values, because you'll know what you're getting and you'll know what the likelihood is of that male passing those important traits onto his offspring. This is just a figure depicting the EBVs of two different ram sires. So if you look at both of them, if you were to breed both of these rams to your ewes, you would expect if one was plus 0 EBV and one was minus 50%, if you bred both of them to your ewes, then you would expect that the fecal egg counts of the lambs that were sired by ram A would be 25% lower than fecal egg counts of those lambs that were sired by ram B. This is a very effective and proven tool, and so whenever possible, you should be purchasing rams that you have these values for, or rams and bucks that have been in a performance test and have some data on fecal egg counts that you could look at and evaluate their parasite resistant traits. Because as we mentioned before, they contribute 50% to most of our flock and or herd. In terms of selecting buck kids and ram lambs, if we want to select a sire on your own farm from your own kids or lambs, then you would select and collect fecal samples from your top performing or the best 5 to 10 that you like, ideally if you have a number of them, and you would deworm them to get them all clean and a similar starting point. Then collect their FOMAGA scores and fecal samples to measure their fecal egg counts at different time points, and then you could truly evaluate their parasite resistance, and you could calculate your own basically estimated breeding values, because this takes into the estimated breeding values that say NSIP calculates, that's based on a number of different things, but you could sort of calculate a value on your own and be able to choose those animals that are more parasite resistant. So some takeaway points from today: genetic selection for parasite resistance, that's one of the most promising means to control worms in a flock or herd. Some of you might already be doing it and you don't even realize that you're doing it, but if you're deworming based on FOMAGA scores or other performance indicators and you're selecting based on those, then indirectly you're selecting out, or you're culling out those animals that have higher fecal egg counts in your herd. The true measure of parasite resistance is going to be fecal egg counts, and doing just FOMAGA scores, you are selecting both parasite resistant and resilient animals. And so fecal egg counts is good to support, to tell those animals that are truly parasite resistant. And as I mentioned before, I'm one of those that advocate for parasite resistance over resilience. Resistant animals, especially resistant sires, are going to lead to lower fecal egg counts and FOMAGA scores in your herd or flock. And when you have resistant animals, pasture contamination is reduced in your herd. So again, overall, you'll see less frequent deworming, and you will have reduced death losses due to parasites if you're selecting for a parasite resistant herd. So I know over the last couple of weeks we've given you a lot of tools that can be used for effective internal parasite control on your farms, but we just want to emphasize that genetic selection is an important tool and something that everyone should be using on their farm, especially when you're purchasing or selecting that sire who's going to be contributing 50% to other genetics to your herd or your flock. And that is my presentation for today.